días a todos, bienvenidos a nombre del Comité de Seminarios. Al seminario de hoy tenemos el gusto de tener el doctor Eric Scheffer. Ahorita voy a pasar al inglés para poderlo presentar. Welcome everybody. We are here today for today's seminar, for Friday's seminar at CEO. And our guest today is Dr. Eric Scheffer. Let me read a little bit of its bio information of his CV. So the doctor is a physicist and he studied at the University of Stuttgart in Germany and also at the University of Massachusetts Amherst in the USA, where he also received his Master of Science. He did his doctoral studies in polymer physics and chemistry at the University of Constance in Germany and at the University of Groningen in the Netherlands. For his postdoctoral research, he studied a bit of biology at the Max Planck Institute for Molecular Cell Biology and Genetics in Dresden in Germany. As far as regards the prizes he's received the Emmy Noether Award in 2007 that allowed him to establish an independent research group at the Technical University in Dresden. In 2010, he was awarded the ERC Starting Grant, and in 2016, a prize for bold science, this sounds interesting, from the state of Baden-Württemberg in Germany. He is now a full professor of cellular nanoscience at the University of Tübingen in Germany, and this is since 2012. So I give him the floor now for his presentation.
Good. Thank you very much for the introduction. And maybe to put everybody on the same map, I presume that you all know where Germany is, but maybe not where Tübingen is. So Tübingen is in the southwest of Germany, and the university is very old. It's a beautiful old town, and the university was founded a few years before Christopher Columbus sailed to the Americas. And maybe a little bit of trivia. So DNA was actually discovered here in Tübingen in 1869. So not the structure, but the molecule itself was discovered here. And for today's talk, what is interesting is that Johannes Kepler, who is famous for his laws of planetary motion, he was a student at the university here. And Johannes Kepler was also the first one to notice that the sun or that light exerts a radiation pressure. And so 2 years ago, I was lucky to take an image from my house of the comet Neowise, and maybe you don't see it all that well. My camera wasn't all that good, but if you sort of zoom in, you can reasonably well see the comet. And Kepler noticed that the comet tail always points away from the sun, so independent of the position, and then he concluded that the sun must exert a radiation pressure. But I'm not interested in celestial mechanics, but in cellular mechanics. So to be more specific, I'm interested in how cells work mechanically, or the individual molecular machines, how do they work to fulfill their cellular function. And mechanical processes are essential for cells. Without them, cells could not survive. For example, during cell divisions, the chromosomes shown in black here are attached via the so-called kinetochores shown in green to growing and shrinking microtubules. So these are filaments, and then molecular machines generate forces that pull the chromosomes apart. So this is a vital process where mechanical forces are important. Another very important process is occurring in your brains, hopefully this very moment. So in your neurons, there's a lot of, so this is a movie. This would be the cell center of a neuron, and the neuron has multiple extensions, neurons and dendrites. And if you zoom into one of these axons, you can see that there's actually quite a lot of traffic going on. So there's small vesicles, membrane-enclosed packages that are transported, and you can see that it's sort of like on a highway. There's traffic in both directions. There's stop-and-go movement, and it looks rather chaotic. And this is necessary because things need to be transported over large distances where diffusion would not be sufficient. And so this active transport is mediated by so-called molecular motors, or little machines that have their tracks that they walk on. And the image that you see here is diffraction limited, so optical microscopy does not provide you more details on the molecular machines themselves, and in the complex cellular environment, it's sometimes difficult to dissect what the individual machines are actually doing. So our approach is to purify the individual components of this transport system and then characterize it by let's see. Oops. Going backwards. Sorry. We characterize these molecular machines by single molecule force measurements and localization measurements. Now, if you want to see more details, you can use electron microscopy. And so this is an early image of a cross-section through such a axon, and you see a microtubule. These are these filamentous structures that were shown in red on one of the previous movies. They have a diameter of about 25 nanometers. These are the vesicles that are transported. These vesicles are on the order of 100 nanometers in diameter, and they're connected via small cross bridges, they were originally called, and these are molecular motors that are engaged and transport these motors. The microtubules themselves, they sort of act as a scale bar, and maybe you can see it. I have a 3D printed model of it. These are really hollow cylinders and made up of individual tubulin monomers. This is increased about 10 million fold in diameter, and so these individual red and orange colored spots are the individual tubulin monomers of 4 nanometers in size. And these molecular machines, these kinesin motors, they walk along this microtubule. And one of the things we're interested in is what actually limits this transport ultimately. And you could imagine that if there's an obstacle or there's a very large force acting on these vesicles, that the motors would just let go, but this is actually not what is happening. There's obviously many factors that limit transport, how many motors you have, how effective they are, and so forth, what cofactors there might be, but ultimately they're limited by detachment. And so to understand how they detach, we first need to know and understand how the motors walk, and then we can address the question of how the motors detach. And it's not like they just would let go of their track. So this is a static image in electron microscopy. And so if you want to sort of get some idea of how these motors actually look like and work, you can use structural information and then animate the system. And so this is a fairly famous animation of how this kinesin, so-called kinesin motor, looks like. I like to call it a Hollywood animation, yet it's rather beautiful. And so you can see that this motor has two so-called heads. You may want to think of them as feet, and they walk on the microtubule that I've just introduced, and they carry these vesicles along. So if we now purify these components, we on the one hand use fluorescence microscopy, and there is an example here of rhodamine labeled microtubules that are observed via fluorescence. So we use total internal reflection fluorescence microscopy, and these are purified kinesin motors, and they have two green fluorescent proteins, GFPs, attached to their tail. And so these are individual molecules that you see that are translocating along the microtubule lattice. To do so, they need to have a fuel, and the fuel is called ATP, adenosine triphosphate, and so they cleave off one phosphate, and the free energy from the ATP hydrolysis is the energy that they chemical energy that they directly convert into mechanical work to transport, for example, these vesicles that you've seen before. Now, from these kind of movies, you can learn how fast these motors are and how far they walk without dissociating. However, what you cannot measure is how much force they can generate. And to measure forces, we use so-called optical tweezers that were invented by Art Ashkin, and this is his Nobel Prize winning figure 1 of his first paper on optical tweezers, and he could show that by in a focused laser beam that you can hold on to particles. And I want to point out here that the forces that are generated for small particles, they scale with the volume of the particles. He could show that, and also with the refractive index difference. And I'll come back to that. And so how can we use the optical tweezers to measure forces? So here's my own schematic. So we use the purified components, we grow microtubules, we immobilize them on a surface with antibodies, we immobilize the motor proteins on the probes that we trap, typically microspheres, and then we add the fuel, the ATP, and then these motors start to walk along the microtubule lattice, and this motion, you can think of the microsphere as a small lens, and the lens will deflect the laser beam, and this deflection we can precisely measure on a detector. Often it's a quadrant photodiode that we can measure relative changes in the laser position. And how this works, I will explain you in detail as we go along. So as a very brief outline, so I'll explain you the basic physics to put everybody on the same level, how these tweezers work, and then go into more details of how these kinesin motors actually walk and detach. So if you think of a microsphere and just a single light ray, you know from Snell's law that if the microsphere has a different refractive index than the medium, it will be refracted, and as the light ray leaves the microsphere, it will be refracted again. If you think of the light not as a wave, but as of a photon that has a certain mass, and then you know that if a particle has changed its direction, there was a change in momentum. And so if you compare the direction of the incoming ray with the outgoing ray, there was a change in optical momentum. And because of Mr. Newton, we know that there must be a force of equal magnitude in the opposite direction acting on the center of mass of the microsphere. So you can think of this whole system that the microsphere is pushing the light to the left, and the light is pushing the microsphere to the right. So this is a single light ray, and now in the optical tweezers we have a focused laser, and so this is sort of the schematic drawing that Art Ashkin used. So two radially symmetric light rays. I don't want to go into use his schematic drawing, I want to use my own one. I've used sort of an inverted geometry where the laser is coming in from the bottom, and then I place a microsphere in the center. And now I do a Gedanken experiment where I displace it in the vertical direction. So I perform an axial displacement. And then I take a pair of two radially symmetric light rays and I trace them through the microsphere. And now if I compare the the ingoing. So this is the ingoing with the outgoing light ray. There's a change in optical momentum in this direction. It acts on the center of the sphere in the opposite direction. Now for the other light ray, this is the ingoing light ray. This is the outgoing one. And so there's a change in optical momentum in this direction. It acts in the opposite direction on the microsphere. If you add up these two force vectors, there is a net force displacing the particle back downwards. And remember that initially I displaced the particle upwards. So this force, which is actually called the gradient force because it always points towards the center of the optical trap where you have the highest intensity, so the gradient is pointing in this direction. So these are the so-called gradient forces. Now, not all of the light is refracted. There is some light that is reflected, so these are indicated by the green arrows, and you do the same as before. They exert a radiation pressure along the propagation direction of the laser, and this is the so-called scattering force. These two vectors add up, and now in order to achieve a stable trap, the gradient force must be larger than the scattering force, in particular in the direction of the laser propagation. I will try to switch my screen here quickly, and I have a little Java applet that I hope that is working. I'm not sure. Can you see the little microsphere here? Okay,
yes,
excellent.
We can see. Thanks.
Excellent. So it's the same geometry in essence, and so now in this case the microsphere is directly in the center, so you have normal incidence, so there's no refraction, there's only reflection, and there's basically straight transmission, and in this case the particle is just being pushed along the propagation direction. Now there is one, so here in this case the scattering forces are larger than the gradient forces. So there's one additional thing that I need to do. I need to change the numerical aperture, so the incoming angle of the light rays. And now if I do the same thing, now actually the gradient forces are larger than before. So if I displace it upwards, it's being pulled down. This is what I've shown you before. If I put it down, it's being pulled up. I can also push it left or right, and so the optical trap is always recentering it. And so let me go back to my presentation. Good. So that this works, you can see. So one additional point is that this is roughly drawn to scale, so that's a laser focused diffraction limited focus, and so the diameter here of a 1064 laser would be maybe 800 nanometers in water with a micrometer sized particle. Your particle size is typically of the size of the wavelength itself, so geometric optics does not work, and Rayleigh scattering also does not work. So you need to use Mie theory actually to do an exact calculation. If you go to the Rayleigh regime, then the gradient forces scale with the volume of the particle and with the refractive index difference, and the scattering force increases with the volume squared and the refractive index difference squared. So that this works, you can see in this little demo movie where I've trapped a rather large particle, and there is a number of particles that are flowing past it. And optical tweezers are Hookean springs, so the force is proportional to the displacement. You can see here that the displacement is not constant. The reason for this is that the flow speed is not constant. And so in this case, the optical tweezers, the optical force is balancing the hydrodynamic drag force. What are the maximum forces that can be generated? So if you increase the flow speed here, at some point the particle would escape the optical trap. You can make sort of a rough estimate that the maximum force is given in essence by the power of the light divided by the speed of light in the medium. And then there's a prefactor, which is some trapping efficiency, which is usually not that good, maybe about 10% for typical materials. And then if you plug in some reasonable numbers, 250 milliwatts in the laser focus and 10% laser trapping efficiency, then you end up with about 100 piconewton, so 10 to the minus 12 newton. This is about the maximum force of a typical optical trap. Now, this is trapping microspheres, but you can trap all sorts of different particles, including live cells. And you can, if you sort of want to test how good your positional resolution is, you can immobilize particles on a surface and then scan with your laser through such an immobilized particle, either in the vertical direction or in the lateral direction, and then record the position as a function of time. And these are 2 angstrom steps that we could measure here, and there's a water molecule drawn to scale here on the right-hand side. So our setup, in essence, has angstrom positional resolution. The smallest force we have measured is about 1 femtonewton, and the highest force is about 1 nanonewton, factor 10 higher than the estimate that I showed you before. We used special anti-reflection coated titanium particles to reach that limit, and the time resolution is very good. We're not limited by the number of photons. We have very good temperature stability in the setups, and also we need to isolate the systems well from mechanical vibrations. So what I was alluding to before is that you not only can trap inert particles, but you can also trap live cells, in this case bacteria. This is an early movie from Steve Block's laboratory, one of the pioneers of applying optical tweezers to biology. And so you can trap it, you can move your optical trap in all directions, and then once you turn off the optical trap, the bacteria just keeps on swimming, so you do not harm the cells. Obviously, if you crank up the laser power, you can opticute these cells. So in general, we can trap any dielectric particles, maybe from a lower limit of about 10 nanometers. The upper limit is eventually set by gravity, and these small spheres or microspheres are typically called beads. You can also trap small metallic particles. However, you cannot use them for biological measurements because of the absorption, and they do get very hot and would denature the proteins and boil them. And as you can see here, you can trap bacteria, yeast cells, red blood cells, viruses, or organelles within cells for manipulation. Good, so much for an introduction to optical tweezers. Now I want to come back to how kinesin motors work on microtubule filaments. So in essence, the motor interacts with the microtubule lattice, and the center of mass is moving by 8 nanometer steps per every ATP molecule that is hydrolyzed. And the 8 nanometers correspond to the periodicity of the microtubule lattice. So if you look at the structure, it has a regular lattice, and these motors actually walk on these so-called individual protofilaments. And you can see, so this would be a typical differential interference contrast image of how such an experiment looks like under the microscope. This line here is a microtubule. They are 25 nanometers in diameter, and so there's rather poor contrast that you get from them. And this is one of these microspheres, much larger and scatters much more light, so that's why you see these interference fringes. And this is being transported by one of these motors. Now, if you now follow this motion with the optical tweezers and use the quadrant photodiode for detection. Then this is the position as a function of time. This is now on the nanometer length scale. I've drawn a grid here of 8 nanometers, and you can see these individual steps, that the center of mass steps that the motor is taking. The duration of the steps is varying. It's a random process, so you expect an exponential distribution in the duration or dwell times of these steps. Now, in this case, we actually had a so-called force clamp where we followed the motion of the motor and kept the force constant. If you keep the laser stationary and measure the position as a function of time, I told you that the optical tweezers are a Hookean spring, so the force is proportional to the position. And so in this experiment, as the motor pulls the microsphere out of the trap center, it slows down. You can again see these individual steps. And as the force is increasing, steps get longer, and the maximum force that the motor can generate is about a little bit more than 5 piconewtons. And from this experiment, you can already do sort of a back of the envelope calculation of the efficiency of the motor, which would be the maximum load of about 5 piconewtons times its step size of about 8 nanometers. This is the mechanical work that you've performed, and the amount of energy that you have available for it is the free energy of ATP hydrolysis, which, if you then do the math, is about 45%, which is quite good. What is remarkable about these molecular machines is that they can directly convert the chemical energy into mechanical work. There's no intermediate step of electricity or heat or something like this. Good. So now let me come back to this schematic of how these motors walk. Now the question is, you can think of these microtubules actually as a 13-lane highway. So each of these lines here is a so-called protofilament, and these kinesin motors, they interact specifically with one of these tubulin monomers. So you can think of it, or can ask the questions, do these motors actually walk in a straight line on a single protofilament? Do they switch protofilaments? And if they do so, is there a directional bias to it, or is it randomly switching them? And to address this question, we actually looked at another kinesin motor. And so you have 14 different types of kinesin in your body, and they all have different mechanical function. The kinesin, conventional kinesin 1 that I told you about before, is involved in vesicle transport. The kinesin that I'm showing you data on now is a kinesin 8, and this is involved in microtubule length regulation. So here you can see that if you add many motors to a microtubule, these motors actually walk to the microtubule end and then act like a Pac-Man and chop off the end of the tubulin and then thereby shorten the microtubule. And so if you sort of, so we asked the question, how does it actually get to the end? Because the conventional kinesin usually only walks for takes about 100 steps, and then it dissociates from the microtubule lattice. 100 steps is about 800 nanometers, or about 1 micrometer. You can see that these microtubules are, this is a scale bar here, so they're inside cells. They're typically more like 10 micrometers. So if the kinesin motor lands somewhere in the middle of the lattice, it would not make it to the end. And so we asked, why is this motor actually able to get to the end? And so when you look at single motors, this is actually the movie that I showed you in the beginning. This motor can actually translocate along the whole lattice of the microtubule. And so it must be very processive. And we looked into this, and so there's a second microtubule binding domain, and there's an interesting feature of it, of the motor, that I'll come back to. Now, the other requirement to get to the end is actually inside a cell. A microtubule is a crowded environment. There are lots of motors and other proteins that are interacting with it. And so if it would just walk on a single protofilament, it would could bump into an obstacle, and then it would also not get to the end. And so to address the question whether these motors can switch lanes, in essence, we devised an experiment where we fabricated a topographic structure, and we suspended microtubules on top of it. And then we have developed a force clamp, or a feedback, where we can trace the microsphere in 3 dimensions, that the motor, we can trace the motor around the microtubule. In the image, this looks like this. So you have these topographic structures, and then these particles, we can trace them along the microtubule as they walk. And so this is now some data. So if you look from behind on the motor, or maybe let's look at this graph here in the middle, the kinesin 1, the transport motor, is actually known to trace a single protofilament, and actually walks in a straight line. Now, the kinesin 8 motor is not walking in a straight line, so you can see that it's actually switching directions multiple times, and it's actually switching protofilaments. If you look at the angle as a function of time, it does not change for the kinesin 1. And for the other one, it's changing directions. If you look closely, you can actually also see angular steps, and the step size actually corresponds to the angle that you would expect if you from the 13 protofilaments that you have. So we think that the motor is actually able to bypass obstacles by switching protofilaments. Now, this motor is very interesting because it's actually rather weak. I told you that the kinesin 1 can generate 5 piconewtons of force. This motor can only generate about 1 piconewton of force, and then it actually starts to slip backwards. You can also see that it's taking backward steps under load, and it always often slips backwards. And we know that it's actually not detaching during these events. And so this is sort of a molecular safety leash that tethers the motor to the microtubule lattice, and that actually explains part of its processivity, why it can actually take so many steps without dissociating. And being a physicist, we actually asked the question, is there any friction involved as you slide one of these proteins over the surface of the microtubule lattice? And so what is friction? So friction resists the relative motion of two bodies in contact, and so you can imagine that the contact on the molecular level is mediated by weak adhesive bonds between the molecule and its track, indicated by these sort of small white lines. And now, if there is a force that slides this object or the objects relative to each other, you deform these contact sites or these adhesive bonds, and then you rupture the bonds, and the energy that you put into the bond deformation, this you cannot recover, and this is the molecular origin of dissipation and friction. And so for the kinesin and the friction or the drag coefficient is related to the diffusion coefficient via the Einstein relation. So this is the drag coefficient gamma is equal to the thermal energy kT divided by the diffusion coefficient. And we discovered that this motor, kinesin 8, if you don't provide it with its fuel ATP, but only with the hydrolyzed version ADP, adenosine diphosphate, it's actually not moving in a directed fashion, but it's actually diffusing back and forth. So our idea was that we use now this motor as a model system to look at protein friction. So we can track the individual molecules, and from the data analysis, from mean square displacement analysis, we can determine the diffusion coefficient. Then we can use our optical tweezers to grab onto one of these particles and drag them over the microtubule lattice and measure the drag coefficient independently. And then check whether the Einstein relation actually holds. And so these are 3 molecules that are diffusing on the microtubule lattice, and then we calculated the mean square displacement as a function of time, and from the slope is 2 times the diffusion coefficient, and so here we can determine the diffusion coefficient, and then when we grab onto these particles or the motors and drag them, measure the friction force as a function of the position where we drag them over the microtubule lattice. There's an initial transient response when the motor is binding, and then there is actually a constant friction force that you actually expect, much like macroscopic friction when you slide 2 bodies relative to each other. And we've tested this as a function of the pulling speed, and the average friction force increases with the pulling speed. And from this relationship, we can then extract the drag coefficient, and then we found that actually the Einstein relation also holds for these single molecular machines. So there is friction, protein friction in our bodies. You all know the effect if you work out that your muscles get warm. This is due to protein friction. And good. Now I want to come back to actually some details of how these motors actually walk. So these molecules, proteins, are so-called heterodimers. So there's 2 identical protein chains that are intertwined, and they form a dimer, and so they walk on the microtubule lattice. And you can think of them as 2 feet, and they pretty much, in this Hollywood animation, you saw that they pretty much, it looks like they walk like we humans do. However, first of all, they don't have a hip joint, so they would waddle more like a penguin, maybe. The second thing is that they don't have a left and a right foot, but they either have 2 right feet or 2 left feet. And so if your right foot is actually taking a step, there's a rotation associated with it. There must be actually a 180-degree rotation, and if that would be, for example, counterclockwise, your next foot should actually take also a counterclockwise rotation. So you would actually expect that the motor is rotating and not walking like we humans do, but more like making a pirouette. Usually in person I would like to demonstrate this, but this is a bit difficult online. So there was an early hypothesis by Joe Howard, and I want to read it out. So the model makes an unusual and seemingly unthinkable prediction. Every step is accompanied by a 180-degree rotation. The rotation will always be in the same direction because of the assumed equivalence of the heads. Thus, the tail and organelle will tend to wind up like the rubber band of a toy airplane. Well, this hypothesis was tested and people did not see these rotations. I wanted to revisit this problem by actually developing very sensitive rotation probes that we can actually simultaneously measure translation and rotation. So to this end, we made birefringent microspheres out of liquid crystals, and our trapping laser is linearly polarized, and so the birefringent microspheres, they align with the polarization direction of our trapping laser. Now, if these motors rotate, they should exert a torque on these birefringent microspheres and rotate them, and if they're rotated, they will change the polarization direction of the trapping laser, and this is what we used for detection of the rotation signal. And so we added an additional detector for measuring angles, so an optical microprotractor, and we operated the tweezers as a torsion balance, and then what we found is that, again, the displacement as a function of time in blue is the translation, and again you can see these individual steps, and in red now is the rotation and signal, and the rotation signal, so there's concurrent steps that always, if there's a translational step, there's a rotational step, and the rotational step is always in the same direction, which implies that the motors actually do rotate in the same direction, always. And the motors also generate a significant amount of torque, and so what this means is that they perform rotational work, and if you add the rotational work to the overall efficiency of the motor, so for the translational work, this was about 45%, and here you add another 35%, and so this gives you about an efficiency of about 80% of the overall motor. So we think that kinesin works with a so-called rotary hand-over-hand mechanism. Now, the last but not least, and I already noticed that I'm a little bit running late, so I'll try to speed up a little bit. There was another controversy in the field whether the motor is actually taking some substeps. So during an ATP hydrolysis cycle, there were some early reports that there might be some substeps in here. But then there were later measurements that showed there were no substeps. So this was controversial for more than 20 years. And so we were wondering what determines our ability to be able to resolve small, fast steps. And so if you think of an individual step as a function of time, it will have a certain step size, delta L, and a certain duration, the dwell time. And it turns out that the product of the step size multiplied by the square root of the dwell time must be larger than a certain constant. This makes sense. So if you make the step size larger, it's easier to detect the step. And if you make the step longer, you can average more over it and thereby reduce the noise. And this reduction in noise is with the square root of the number of independent data points that you can average over. The constant itself does not depend on the experiment, really. So you can think of this as being some sort of uncertainty principle for molecular step detection. It, in essence, depends on the thermal energy, kT, and the thermal motion, in essence, the drag coefficient of the particle. And so this is a constant, and if you think of this relationship, because it is a constant, I can just plot the step size versus the dwell time, and I expect an inverse relationship. And so this is the step size as a function of the dwell time on a log-log plot, and this is sort of the square root inverse relationship, and anything that is above here is observable. And these are some of the measurements that people have done of other molecular machines. This is the so-called RNA polymerase that takes one DNA base pair steps, so 3.4 angstroms. This is a DNA packaging motor. This is kinesin, the original study where it was actually slowed down, so 8 nanometer steps slowed down to 1 second per step, and this is myosin. There are no data points in this black region down here. So the question is, why are there no data points here? There's no, so you can get there by making smaller particles, but why don't people use smaller particles? Well, here I come back to the statement that the gradient forces, they scale with the volume of the particle. So if you make your particle 10 times smaller, forces decrease by a factor of 1,000. And then forces are not sufficient to counteract the forces that are generated by the molecular motors. You could increase your laser power by a factor of 1,000, but usually you don't have that much laser power available. And if you would have that much laser power available, you would actually have a little bit of heating, or you would have heating which would be too much. Now, our idea was to actually use the other scaling relationship that is proportional to the difference in refractive index and use the highest refractive index materials to compensate for this force volume scaling. And so we looked up materials, and germanium actually has the highest refractive index. So we first performed some mean theory calculations of the trap stiffness as a function of diameter, and this is the trap stiffness normalized by the laser power. And so this red curve shows you that for germanium we have which has a much higher refractive index, we have a more than 10-fold higher trap stiffness compared to silica. Now, unfortunately, you cannot buy germanium particles, and so we actually ended up developing our own synthesis method, and we were successful to measure or to fabricate size-controlled particles, and we used X-ray diffraction to confirm that we actually do have germanium particles. And then we put them back in the trap and measured the trap stiffness. This is the data, and they fall perfectly on top of the calculations, and there's no fit parameter in here. If I plot it on a log scale, you can see the volume scaling of the trap stiffness. So there's 3 orders of magnitude here in the trap stiffness as you increase the diameter by 1 order of magnitude. Good, so we decided for the experiments of the motors to use 70 nanometer diameter particles, and then we put them in the trap and first tested whether we can measure the trap stiffness that we needed, and we record the position as a function of time, and then for calibration purposes calculate the power spectral density as a function of frequency, and you expect a Lorentzian type response, and this is a comparison of about 600 nanometer polystyrene particle at the trapped with the same trap stiffness, and you can see this, and this is the 70 nanometer germanium particles. These schematics are drawn to scale, and you can see that the noise level here in nanometer squared per root hertz goes down as you have smaller particles. So this is an improvement in spatial resolution. On top of it, there is this characteristic roll of frequency that gives you a measure for the time resolution that you have in your system. And this increases by about a factor of 10 here. And so this characteristic roll of frequency is related to the time resolution that you have, and for these germanium particles we have about a 10 microsecond time resolution for the measurements. So for the experiments we then put a lipid bilayer around it and coupled these motors through their fluorophore and the so-called GFP nanobody to these particles, and they are in size pretty much correspond to the size of these transport vesicles, and this is the microtubule, so this schematic is roughly drawn to scale. That these particles are being transported properly. You can see in this interference reflection microscopy image of a microtubule, and this germanium nanosphere is shown up with the white contrast. Now, if we put these particles in our optical trap and record again the position as a function of time, if you look very closely, each of these steps, there is an intermediate mechanical step, and we can use a step detection algorithm to quantify these. And then if we plot a step histogram, the motor actually steps with 4 nanometer substeps. There's a small proportion of 8 nanometer steps. These are fast steps that we probably cannot, 2 fast steps that we probably cannot resolve individually. I want to draw your attention to the last step, which is often one of these. So you can see that there's always one long step and then there's a short step, and the last step is often a short step. And then, similar like with the kinesin 8 motor, I would like to zoom in and show you now that the motor is actually not detaching but slipping. So what do we expect for detachment? Then we would expect an exponential relaxation back to the trap center. If we zoom in now to the microsecond time scale, we can see an exponential relaxation. However, if we quantify this, it's about 300 microseconds, and this is much higher than the time resolution of, or the relaxation time that we actually expect in our optical trap. So if we pull perpendicular to the microtubule axis, then we can actually measure this a much shorter relaxation time, about 30 microseconds. This is roughly what we expect in the vicinity of the surface. So if we pull parallel on the microtubule axis, there's an increased drag coefficient. The trap stiffness kappa is constant, and so where is this additional drag coming from? Well, it's coming from protein friction. And so since we know all of these parameters, we can actually calculate how large the friction is. We can use the Einstein relation to convert it to a diffusion coefficient, and we can take it one step further and use a random walk model and actually calculate that we would expect diffusional steps of duration of 70 microseconds. Now, if we use a more fancy step detection algorithm than our own, we can actually detect individual slip steps that we call, and they actually have 70 microseconds duration and about a size of a little bit less than 8 nanometers. And so in none of the traces that we had, we've observed this very short relaxation back. So the motors were always attached, still attached to the microtubule lattice, and they never let go directly. Why is this important? Well, to come back to this vesicle transport scenario, it's not transported by a single motor. There's usually a small team of motors that are transporting them. They're random steppers, so there's always one ahead of the others. And so if one is actually lagging behind too much, it can slip and then catch up with the others. Or if one is too far up, it can slip back to the team. And this is sort of enhances the overall transport. So I would like to wrap up and sort of tell you how this motor is walking. So this is this Hollywood animation. This is what is called the asymmetric hand-over-hand mechanism, pretty much like we humans walk. Now, if you add the rotation to it, this is the rotational or rotary hand-over-hand mechanism. And then now we also need to add the mechanical substep. So there is always a 4 nanometer step in between. And this is now how we think that this motor is walking. I should point out that here on the left, this is Swati. She has performed all of the germanium nanosphere experiments, developed the synthesis, and performed all of the experiments. So we revised this Hollywood animation. There's still quite a bit of Hollywood in it, but conceptually this is closer to the state of the art now. So the heads are always rotating in the same direction, and then there's an intermediate step in between as the motor is or the vesicle is being transported along the microtubule. And so if we now come back to this graph of the step size versus the average dwell time, if we put now these are the 4 nanometer steps, they have they're a few millisecond duration, and they're slightly below this line of what has been previously measured. So maybe some labs were actually able to see some substeps, and other labs did not have the sufficient resolution. So we think that may have been the reason why it was controversial and people did not see these substeps. Now these very fast slip steps, there are 70 microseconds in duration and closer to 8 nanometers. They show sort of the limit of what we can achieve. And so drawing the line through here, this corresponds in essence to a 4.5 improvement in spatial resolution and about a 20-fold increase in the time resolution of optical trapping experiments. And I think this opens up new possibilities for other molecular machines. If you take the analogy to microscopy, for the resolution limit limited by diffraction is for a good microscope maybe about 200 nanometers. If you divide this by this factor 4.5, you would have a resolution of about 45 nanometers, which is pretty good for super resolution microscopy. And so this brings me to the end. First of all, I would like to acknowledge Art Ashkin for inventing the optical tweezers. I was lucky enough to meet him a few years ago at this conference where he actually gave his Nobel lecture for the official Nobel ceremony in Stockholm. He was actually could not attend it, and so he gave his Nobel lecture at this conference. There's a link down here. So if you Google Nobel lecture Art Ashkin and Aspen, you will probably find it. It's actually quite interesting, and it was really inspiring to meet him and discuss his science, for example, during World War II on radar technology and so forth. And so it was really inspiring. And at this time, he was actually actively still working on a manuscript on harvesting solar power for energy. So he was still active to at the age of 96. Last but not least, I would like to thank my team doing the experiments, in particular Schwati for the germanium nanospheres. These are my collaborators and funding sources, and I would like to leave you here with a summary slide of what a German could do with germanium nanospheres. Thanks.
Wow, thank you so much. It was really nice to listen to this story. So let me check if there are questions in the chat, or also everybody can take the microphone and ask directly a question to our speaker, or if you prefer, you can write it down either in Spanish or in English, and I can read it directly to our speaker.
Okay, so in the chat there are a couple comments in Spanish, which unfortunately my Spanish is not good enough for that.
Yes, actually they were talking about well, there is one question. Let me read this one. This is the first one, actually. The other ones are technical communications. Thanks a lot for the talk. It was fascinating. In the graph of the steps, does the fact that in the periods of the time of the torque the torque comes back to its initial value mean that it unwinds also? And could you also analyze how this relation takes place?
Yes, so that refers to our rotation measurements. So the question refers to this quick movement backwards, where indeed the torque is being released, and there's an implication for the walking mechanism of how they can even generate the torque, because in the original Hollywood animation, the motor is actually lifting its feet off of the microtubule lattice. If you lift the heads, then the motor or the cargo is only connected through a single polypeptide chain, and that is actually free to rotate. So our measurements actually imply that the two heads are always in contact with the microtubule lattice, and we actually think that they're electrostatically guided along the microtubule lattice. Now, in this scenario here, we think that actually one of the heads is indeed truly detaching, and then the torque is released through a swiveling mechanism of this single polypeptide chain. I hope this answers your question.
Okay, well, she will probably let us know. There is another question here. Do you know if the laser power influences the longitudinal position of the trap? That is, if the optical elements involved in the formation of the trap cause any change in the longitudinal position of the trap when the laser power is changed? I mean, just the trap without any particular particles trapped.
If you change the laser power without any particle being trapped, then it's It depends on your laser. Let's put it that way. I mean, we have a solid state laser, and so there in the cavity there's a thermal lens, and if that depends on the laser power, then there might be a pointing fluctuation or a change in the position. In our system, we always keep it at the maximum power, and well, maybe I can actually go. I have some. This is sort of a rough schematic of one of our setups, and in essence, this is a 5-watt neodymium yttrium vanadate laser. It's linearly polarized. There's a Faraday isolator beam expansion here, and then we have a half wave plate that rotates the linear polarization. And we, together with the orientation of this polarizer, the orientation of this half wave plate determines how much we transmit and how much we dump. And so the laser is always operating under the same power conditions and only through the rotation. And we align all of these optics that through the rotation, the beam position does not change. In general, if you do have a particle trapped, then the trap stiffness changes as a function of, if I go back to the data here, so this is the trap stiffness per laser power. So this is normalized, and so the trap stiffness increases linearly with the laser power. So if you have twice as much laser intensity, your trap stiffness is twice as high. If you don't have any heating effects or other, if you're the beam quality, so it's if the beam shape does not change, and in the so does that answer your question?
So while we wait for the answer and we wait for other questions also, I would like to ask something myself. I think it's absolutely interesting, everything you have introduced, and I'm particularly curious about the use of germanium nanosphere for the study of, other motor proteins. Have they become the preferential tool for doing these studies, or are they becoming like that? Are you selling them, or?
It's not yet, and we're actually struggling ourselves to make the particles. Shwati has left the lab, and we have actually trouble making them again. And we look into the chemistry, and it's actually rather complicated. And there's many competing processes occurring here at the same time, and they're also not stable in water. So they convert to germanium oxide again, which has a very low trap stiffness. And so you always need to make them fresh, and that is a bit of a drawback, that they're chemically not that stable and that the synthesis is not straightforward. And so we can reproduce it, but not on a regular basis. So a lot of people have asked me for samples, and there's no other protocols out there to make such beautiful particles. Silicon would be much better. That would be more stable. At the moment, we're looking into other particles, so-called upconversion particles, nanoparticles that we trap close to their resonance. And there's one paper out there that shows that you can achieve an even higher trapping efficiency with these upconversion nanoparticles, but they have not been applied to any biophysical measurements. And these upconversion nanoparticles, they're chemically very stable. And so I think they're more promising than the ones that we have at the moment. Unfortunately, I would be very happy if we could easily make them and store them in particular, but we cannot store them either. So the lipid bilayer preserves them somewhat, but then after a few weeks, they go bad again. And it's very the measurements are very tedious because they're very small. You don't see them in the microscope, so you actually need to trap them blindly, and so these are tough experiments, let's put it that way.
I see, I see. So I was actually going to ask you if the behavior of kinesin has been characterized by means of this kind of experiment for other types of kinesins. So you mentioned kinesin 1, kinesin 8. How much do we know about different types of kinesins, and how much do we believe that they all behave the same way or in similar ways? For example, with this rotation, about the heads and so on.
Well, kinesin 1 has been the role model in the field, and so far for almost all of the motors, people have measured these 8 nanometer steps. There's measurements almost for every type of kinesin. The forces vary, the speed at which they walk varies. There's a few strange kinesins or orphan kinesins where there's different step sizes that have been reported, and there the distance between the heads, the so-called neck linker, is longer, that they have a longer reach, that they can actually take longer steps. But this is not very well characterized. And the rotation, just based on so for the ones that take 8 nanometer steps, if it's a homodimer per definition, you would expect these unidirectional rotations. And the reason why people have not seen these rotation previously was that the rotational probes that people have used before, the rotational relaxation time was very large, so that they had to slow down the motor significantly, and meaning they reduced the ATP concentration to really nanomolar concentration, that you have maybe 1 step every second. And if we go down to very low ATP concentrations, we actually don't see rotations either. So we have to be above the KD of the motor, which is about 70 micromolar. So if we're above that, we do see the rotation. And we think that the reason for this is that if you give the motor too much time for a step, eventually it will lift one of its heads or detach one of the heads truly from the microtubule lattice, and thereby release the torque. So it cannot build up torque if it's being slowed down too much. So I think that if the motor is sufficiently fast, that any of these motors should rotate. And this has not been shown because, again, these are also not easy experiments. There's some gold nanoparticle tracking experiments that are consistent with this rotation measurements. There's some min-flux measurements from Stefan's Hell's lab. There's a preprint at least, and they do show rotations, or it's consistent with the rotations as well. But and I think the basic mechanisms, so kinesin 1, I think, does serve as a role model. For the kinesin 8, we have now also measured these mechanical substeps. So the overall hydrolysis cycle is probably very similar for the different kinesin types. It's only how much time it spends in a certain nucleotide state that differs, and the individual affinities of the heads to the microtubule lattice. That is where the variation comes in.
I see. Well, I have one last question, and the previous answers were the people that asked the previous answers are okay with your answers, and they thank you. And my last question is about, I think you have already answered it partially, because you revealed how difficult it is to synthesize this particle, to maintain them, and also to do the experiments. But my question is actually out of curiosity. How far do you think we are from using experiment like yours to understand differences in microtubules? So using the system with the trap and the particle and the motor protein to understand differences in the surface of microtubules. Is this something feasible or something which is still very far away?
No, it's feasible. There are some experiments, or just based on fluorescence with optical tweezers. I'm not quite sure if somebody has looked at, or maybe I should actually go to the animation here. So for everybody, there's these wiggly lines that I've added here in the animation. So these are the ends of the tubulin molecules, and these are unstructured domains, and they're post-translational modifications of these. So they're chemically modified inside the cell, and that determines some of the functionality or which motors are binding. And so people have measured different speeds on different types of post-translational modifications. I'm actually not aware that people have done trapping experiments on it. We have actually performed our friction experiments on subtilisin treated microtubules, where we've cleaved off these C-terminal tails, and we've seen some effect, a modulation or an increase of a factor 2 in the diffusion coefficient and the decrease in friction, but it's certainly feasible, and with now tubulin becoming available also by recombinant protein expression and having different types, this is certainly feasible experiments. In terms of their translocation speeds, there were not factors of 10 difference between them, but more like on the order of 30-40% differences, and people are looking into that. You would have to look at, I'm not sure whether people have looked at the individual nucleotide states, how they depend then on the post-translational modifications. It's certainly feasible, but I'm not aware that anybody has done it so far.
I see. Well, thank you so much. Thank you again for this beautiful talk, and I will soon send you a certificate for participating in this Friday seminar of our center. Thanks a lot, and I think we have finished here.
Okay, thank you very much. Thanks everybody for listening. Looks like there were actually quite a few people here.
Yes, about 100.
Okay, excellent. So thank you very much, and have a good day. For me, it's a good evening now, and have a good weekend. And bye,
you too,
everybody.
take care. Thanks a lot again. Goodbye.
Yeah, you're welcome.